So we were doing kingdom Monera, and the kingdom Monera, as you know, there are two different types, eubacteria and archaebacteria, right? Under eubacteria you will be studying bacteria, cyanobacteria, then mycoplasma, and under archaebacteria we are supposed to study halophiles, methanogens, thermoacidophiles. And we had started with bacteria, the general bacterial structure we have completed, and now we are on the second thing, that is bacterial nutrition. Okay, so let us start with nutrition in bacteria. Other than nutrition, we will be slightly focusing on the reproduction aspect of bacteria as well. So now let us start with bacterial nutrition. Nutrition in bacteria. Bacteria may carry out autotrophic as well as heterotrophic nutrition. That's what you have written in the Whitaker's 5 kingdom classification table. Under autotrophic, photoautotrophic is possible and chemoautotrophic. And under heterotrophic, there are 3 types: parasitic, symbiotic and saprophytic nutrition. So we are going to look at both of them in some detail. Let us start with autotrophic nutrition first. Autotrophic nutrition. What does this mean? Autotrophic and heterotrophic. That means that the bacteria, some of them are autotrophic and some of them are heterotrophic. Heterotrophic, majority of them are heterotrophic compared to autotrophic. Okay, but we are still looking at it. Autotrophic nutrition in bacteria. Autotrophic nutrition may be of 2 types: photoautotrophic and chemoautotrophic. Photoautotrophic and chemoautotrophic. Photo and chemo, that means these use light energy for their glucose synthesis, and they use chemical energy for their glucose synthesis. Okay, so what they do is they use light energy, use chemical energy. Let me elaborate this further. They trap sun's light into ATP, further used for glucose synthesis. Okay, and what about chemoautotrophic? They oxidize certain chemicals, and the energy that is liberated is trapped into ATP synthesis. These trap sun's light into ATP synthesis, and they use the chemicals energy after oxidizing it. Oxidize inorganic chemicals like nitrates, nitrites, ammonia for ATP synthesis, which is used for glucose synthesis. Okay, both of them require ATP for glucose synthesis, but where is their ATP coming from? This guy is coming from sunlight. Sunlight is trapped into ATP, and here oxidizing chemicals. Photoautotrophic, chemoautotrophic. Fine. These bacteria that are involved in photoautotrophic nutrition are called as photosynthetic bacteria. Photosynthetic bacteria are not to be confused with cyanobacteria of eubacteria. Under eubacteria, if you go back to that table, okay, there is bacteria and there is cyanobacteria also, isn't it? So the photosynthetic bacteria that we are learning about are in this category right now under bacteria. These photosynthetic bacteria are not to be confused with the cyanobacteria that you will be doing later. Okay? Why? Because all the cyanobacteria are also photosynthetic. That you know? Okay? All cyanobacteria are photosynthetic. So these are also photosynthetic eubacteria, and they are also photosynthetic eubacteria. But what is the difference? There are differences. So we will look at them right now. These photosynthetic bacteria not to be confused with cyanobacteria. Okay, so cyanobacteria, I will just mark down some differences. So some differences are that the pigments are different. Photosynthetic bacteria have pigments like bacteriochlorophyll, okay, and cyanobacteria have pigments like chlorophyll A. Chlorophyll A is also present in plants also. present in plants, the green plants. Okay, so these pigments are different, bacteriochlorophylls and chlorophyll A. Second thing, the photosynthesis is anoxygenic. What does this mean? Anoxygenic, does not liberate oxygen. Their photosynthesis does not liberate oxygen. Different from cyanobacteria in that their photosynthesis is oxygenic, means it liberates oxygen, liberates O2, does not liberate O2. Got it? It does not liberate O2. Okay, more under autotrophic below this. Examples include photosynthetic bacteria like purple bacteria, green sulfur bacteria. One of the green sulfur bacteria is chlorobium, very popular type of bacteria, and green non-sulfur bacteria. Green sulfur bacteria, example chlorobium, and green non-sulfur bacteria. These are the different examples of photosynthetic bacteria, not to be confused with cyanobacteria. Okay? Not to be confused with cyanobacteria. Very important. Next. So these are photoautotrophic. They use sunlight for their glucose synthesis, and these are chemoautotrophic bacteria. They oxidize certain chemicals like nitrates, nitrites, and ammonia for ATP synthesis, which they use for their food synthesis. Cool? They play important role in recycling minerals like N, P, S, K, etc. And like N, P, S, Fe, etc. This is an important sentence from NCERT. They ask questions from this. Next, examples include organisms like Nitrosomonas and Nitrococcus. These are the examples of chemoautotrophic bacteria. So this was the autotrophic nutrition in bacteria. Now we are moving on to heterotrophic nutrition in bacteria. Okay, now we are doing heterotrophic nutrition. Heterotrophic nutrition in bacteria can be divided into three different types: parasitic, symbiotic, and saprophytic, wherein saprophytic nutrition is the most abundant. Now you know the difference between autotrophic nutrition and heterotrophic nutrition is that in heterotrophic nutrition the organism depends on ready-made food, right, from some source. So based on the source from whom these bacteria absorb their nutrition, they have been divided into these three different types. In case of parasitic nutrition, the source of the nutrition is a living host from which it draws nutrition. In symbiotic nutrition, the bacteria will absorb nutrition from another organism, but give back something back again also to the organism. The difference between parasitic and symbiotic nutrition is that in parasitic nutrition, the bacteria will only keep on absorbing without giving anything in return. In symbiotic nutrition, the organism takes nutrients from the organism, however it gives back also. It's a symbiotic association. And in saprophytic nutrition, the source from which the bacteria derives its nutrition, the source is dead or decaying organic material. Okay, so these are the differences between these three different types of heterotrophic bacteria. Let's write down over here. Bacteria derive nutrition from a living host, not dead. Okay, and cause diseases in the host. These type of parasitic bacteria are also called as pathogens or bacterial pathogens. Okay, whatever. They cause diseases in the host. For example, Vibrio cholerae cause the disease cholera into the living host. Okay, and it just keeps on drawing the nutrition. It's a parasite, it's a pathogen, that kind of a heterotrophic nutrition, and cause diseases in the host. Let's write down examples. Vibrio cholerae causes cholera. Then Salmonella typhi cause the disease typhoid in the host. Next, Xanthomonas citri cause the disease citrus canker in plants, in citrus plants. This is a plant pathogen. Okay, write this down, plant pathogen. Then Clostridium tetani cause the disease tetanus. Right, so that is about parasitic nutrition in bacteria. Symbiotic, bacteria live in mutual relationship with another organism. This is with, this is with, this is which. Okay, short forms. Bacteria live in mutual relationship with another organism. For example, nitrogen fixing bacteria, Rhizobium. Have you heard of them? Rhizobium bacteria, nitrogen fixing bacteria, Rhizobium, live in root nodules of leguminous plants. Okay? Saprophytic bacteria live on dead, decaying organic matter. Dead. Okay, for example, curd forming bacteria, Lactobacillus. So what will Lactobacillus do in milk? So that's its dead organic material. It will derive nutrients from it and secrete byproducts that will be involved in the formation of curd. So with that we have completed the nutrition in bacteria. Moving on to reproduction in bacteria. So this is not there in a great detail in NCERT, so we will just do this very superficially and light, and some depth is there in 12th standard. We will see that in the 12th standard syllabus. Right now I will tell you briefly reproduction in bacteria. Okay, next we are doing reproduction in bacteria. Reproduction in bacteria can be carried out in two different ways: asexual reproduction and sexual reproduction. As you know, in asexual reproduction only one bacterial cell is involved, only one parent is involved, and over here two bacterial cells involved. Okay, two parent bacterial cells involved. In asexual reproduction, one bacterial cell only will produce the subsequent daughter cells. Okay, and over here two different bacterial cells are involved. Okay, let us look at asexual reproduction. There are two different types. One is binary fission, second is endospore formation. In binary fission, one bacteria divides into two daughter cells. So binary fission. Okay, one parent cell divides into two daughter cells. Binary fission is carried out in favorable conditions, when everything is fine. But when there are unfavorable conditions, they prefer to undergo endospore formation. What the bacteria does in unfavorable conditions is that it will produce a thick spore around its genetic material. A thick-walled endospore will be formed around its genetic material. This is called endospore. Unfavorable conditions. Because of the unfavorable conditions, all the bacterial components will dissociate, but the endospore shall remain viable, carrying the genetic material inside. Upon the return of favorable conditions, the genetic material inside, which carries all the genes to produce the structural components of this bacterial cell, it will produce all the structural components around itself and will come out of the endospore as a new bacterial cell, new daughter cell. So one parent has produced another daughter cell, okay? It is a form of reproduction, something has reproduced, okay? That's why it is under asexual reproduction. One parent, one child, one parent is involved, one parent is involved. So it is asexual reproduction, okay? Favorable conditions, unfavorable conditions. This endospore is said to be very, very thick walled, and it is very resistant to unfavorable conditions like high heat, high pressure, etc., wherein the bacterial cells genetic material will sustain inside. It will thrive. The genetic material of the bacterial cell inside the endospore will thrive under very high, very extreme kind of conditions as well, okay? And the bacteria shall remain upon the return of favorable conditions. One parent is involved, one parent is involved, that's why it is asexual reproduction. Moving on to sexual reproduction. Minimum two bacterial cells are involved over here. Now, in sexual reproduction in higher animals, how does the sexual reproduction in higher animals take place? Gamete from one parent, gamete from another parent carrying the genetic material will fuse and will form the zygote inside one parent, and that will grow into the embryo, isn't it? However, over here there are two bacterial cells. One bacteria, they are themselves one-celled parent cells, okay? So even if one parent donates its genetic material to the other parent cell, that parent cell cannot produce a baby bacterial cell inside it, okay? That bacterial cell cannot produce a baby bacterial cell inside it. So this bacterial cell will itself undergo changes, okay? This bacteria will change upon the receipt of that genetic material from the second bacterial cell. Got it? So that's how this has undergone changes. A new bacterial cell has been formed, okay, with new traits. That's why this is not called as sexual reproduction, it is called sexual recombination. It is different from sexual reproduction, okay? It is called sexual recombination, and this is sexual and this is asexual reproduction. There are three different ways how one bacterial cell can take up the genetic material of the other bacterial cells, okay? There are three different ways. One is through surrounding medium in which the bacteria is floating. So some of the bacteria died, its cell disintegrated all the genes outside, they are floating in the surrounding medium, and that's how it takes up and it changes. That is the first method. Second method, wherein one bacteria comes in contact with the second bacteria, okay, and injects its genetic material from one to the other through pilus. You remember pilus of the bacterial cell structure involved in conjugation? So it will donate on itself. Third way, if this bacteria is infected by a virus which has previously infected another bacteria, because when viruses infect one bacteria and when they release out from that bacteria, they will take up the genetic constituents of that bacteria, and when such kind of a virus infects yet another bacteria, then it will donate all its genes to the second bacteria. So that's how three different types of sexual recombination are observed. One is transformation. Transformation is wherein bacteria takes up genetic material from surrounding medium. Second type is called conjugation. Conjugation is wherein bacteria takes up genetic material from who? From another bacteria, through pilus, okay? If you don't remember, it's pilus, okay? It will attach to the second bacterium and it will transfer some parts of its genetic material to this one, okay? Third one, transduction. Transduction is the third type of sexual recombination, wherein bacteria, in transduction, bacteria takes up genetic material from a virus which has earlier infected another bacteria. Okay? In this, bacteria takes up genetic material. Bacteria takes up genetic material brought by a virus which has previously infected another bacteria. That is transduction. So it's like a vector. You remember vector? Asexual reproduction in bacteria is the most common, and within asexual reproduction, binary fission is the most common type of reproduction. So that was all about the reproduction in bacteria.
महादेव के नाम पे सब्सक्राइब कर दें